This podcast is a production of OPENPediatrics, an open access online community of healthcare professionals sharing best practices from around the world. Visit openpediatrics.org for more.
Welcome to the World Shared Practice Forum podcast. I'm Dr. Jeff Burns, Chief of Critical Care at Boston Children's Hospital and Professor of Anesthesia at Harvard Medical School. We're here today to talk about what I would call a landmark paper on MIS-C that was published on August 7th of 2024 in Nature. And I have to say that the alt metrics on this paper right now indicate that this is in the 99th percentile of what's being discussed among recent papers on PubMed. The manuscript is entitled Molecular Mimicry in Multisystem Inflammatory Syndrome in Children. And here with us today are the first author and one of the senior authors. The first author is Dr. Adrienne Bodansky. Dr. Bodansky is an assistant professor of pediatric critical care at the University of California, San Francisco. And also with us today is Dr. Adrienne Randolph. Dr. Randolph is professor of anesthesia and pediatrics at Harvard Medical School and a senior associate in critical care medicine in the Department of Anesthesiology, Critical Care, and Pain Medicine at Boston Children's Hospital. Welcome to you both.
Thank you, Jeff.
Yeah, thank you so much for having us.
As I stated in the opening, it's really reflected in the alt metrics, but of course in the sophistication of the manuscript that we're about to hear about, that this truly is a very significant step in what we understand about mechanism in MIS-C. But before we discuss the paper, I wonder if I could begin with you, Adrienne. And could we talk about really what was understood about the science of MIS-C up to the point of your study? What did we understand about mechanism in this syndrome?
Yes, that's a very good question, Jeff. Just to remind people that MIS-C is a hyperinflammatory syndrome. It stands for multisystem inflammatory syndrome in children. And it emerged early in the pandemic, where initially children were thought to be spared from severe COVID-19, but then this post-infectious condition started appearing where children would present with hyperinflammation, often cardiac involvement, cardiovascular involvement in almost half, and some Kawasaki-like features with rashes and other things, along with dilations of their coronary arteries. That was concerning that this may evolve into aneurysms. Over time, MIS-C has pretty much gone away as the pandemic progressed, but there were over many thousands of children affected globally with the syndrome, and a small percentage, died, and over half were in the intensive care unit. So there were multiple investigators really focusing in on this problem, trying to understand what is triggering this hyperinflammatory condition in these children. Because of the Kawasaki disease-like features, most of the children received IVIG, and about half received steroids as well, or just steroids, and most of them recovered soon after treatment. So people looked at this from multiple standpoints, and because it emerged approximately 30 days to 6 weeks after the initial, usually very mild case of SARS-CoV-2 infection, there was always a question of whether this could be some type of autoimmune trigger. And so people started looking at autoantibodies. And one of the confounders, however, were that a lot of these kids already had gotten IVIG before they drew the sample. And so they had received antibodies. There were some reports of autoantibodies, and Dr. Bodansky here will talk about that because we did look at the ones that were previously reported as well. But still, there was really no clear answer as to what was triggering this in children. We had a few answers in a few cases of hyperinflammatory gene variants, but not in a large percentage of these children.
Terrific. And with that as background, Dr. Bodansky, can I turn to you? What were the specific aims of your study, and what was the approach and methodology that you were pursuing to really elucidate further the mechanism of MIS-C?
Thank you so much for asking me, Dr. Burns. I think Dr. Randolph really beautifully summarized the disease there. And I think one thing to highlight that we really wanted to focus on was this idea that, again, the one thing that we knew with certainty was this disease occurs exclusively following a SARS-CoV-2 infection. And so we really wanted to get at what is the link between SARS-CoV-2 and the subsequent development weeks later of this inflammatory syndrome. And there had been other studies that were indicating that there was immune dysregulation. So alterations in your innate immune system and your adaptive immune system, specific changes in how your T cells were functioning, as well as some studies showing autoantibodies. But I think there were two real limitations in the autoimmune space. One of them was the tools being used to look for autoantibodies, which traditionally require you to have a hypothesis about what autoantibody to look for and then try to test for that. And the other was the amount of controls and which controls were being used. And in some of the cases, there were only 5 to 10 controls that were able to be used. And so what we were able to do is, one, use a technology that was actually originally developed at Harvard in the lab of Steve Elledge, and then was rebuilt specifically for the purpose of autoantibody discovery in human disease in the lab of Joe DeRisi, where I was working at the time, which allows you to look for every single possible autoantibody all at once. And so you can, in an unbiased fashion, look for what's in the patient's blood. And at the same time, we also used the same technology to look for antibodies targeting all parts of SARS-CoV-2, but in very high resolution. So we could look at every little piece of SARS-CoV-2 as if you've chopped up all the proteins into little pieces and get what we sort of would refer to as a high resolution understanding of where exactly antibodies are binding SARS-CoV-2. And then the final piece is we had this beautiful set of controls that Dr. Randolph and her team were able to assemble, which were 45 children who had previously had a SARS-CoV-2 infection, but then had blood drawn 4 or 6 weeks later when they were at risk of MIS-C, and so should have the same background adaptive immune changes, but they did not develop MIS-C. And so what our plan was to broadly profile the humoral immune responses to all host proteins and all pieces of SARS-CoV-2 in the MIS-C patients. So we had 199 of them compared to the 45 controls.
That was a very clear description. This is really terrific. Thank you, Dr. Bodansky. So what do you see as the 3 salient findings of your study?
Thank you, Dr. Burns. It's a great question. It's always fun to try to boil it down to 3 things. So I think the first point is that we did find a unique and specific autoantibody signature in these patients. And so it turned out that children with MIS-C relative to the controls had a number of autoantibodies, but in particular, they had autoantibodies targeting a protein called the ETS repressor called ERFL, as well as autoantibodies targeting a protein called SNX8. And we really focused in on SNX8 initially in our paper because SNX8 happens to be part of an antiviral pathway that had actually already been implicated in cases of MIS-C that had genetic defects. So there's a small set of patients who had been found to have a genetic deficiency in part of the pathway that controls this antiviral response. And SNX8 is part of that same pathway. So we thought, this is pretty interesting. So why don't we dig in a little bit further? And what we did is we looked in higher resolution at what part of SNX8 the autoantibodies were targeting. And it turned out that it wasn't that everyone was making different autoantibodies to different parts of SNX8, but they were actually all targeting the exact same small little piece of SNX8. And so we thought that's interesting. How is this connected to SARS-CoV-2? What if we look again at whether these patients had a different response to SARS-CoV-2 rather than just the human protein? And we found the same thing there. We found that MIS-C patients and controls both make antibodies to the nucleocapsid protein, which people knew, but they actually were making different antibodies. So the typical children were making the typical response to part of the nucleocapsid protein, but children with MIS-C were making a response to a slightly different part of the nucleocapsid protein, but it was a different antibody. And what we did is we did something called alanine epitope scanning, which is a fancy way to say that we found the exact amino acids that the antibodies were binding. And it turned out that the piece of the nucleocapsid protein that MIS-C patients were targeting looked almost identical to the piece of the SNX protein that was being targeted. And this really evokes this idea of molecular mimicry, which is that the virus is having an immune response to something that looks just like yourself, leading to autoimmunity. So I think that's the first really important point of our study, was we found a clear link. I think the next key is that we identified a plausible mechanism for the disease. We didn't prove a mechanism. We have not proven pathology here. That's important to know. But we did come up with a plausible one. And that's that autoantibodies themselves don't typically get inside cells and disrupt processes. And so it seemed unlikely that the antibody to SNX8 was disrupting, a viral defense pathway. However, CD8 T cells, those are the cytotoxic T cells in your body, are often the effectors of autoimmune disease. So for example, in type 1 diabetes, you have anti-insulin antibodies, but those aren't causing the problem. It's actually CD8 T cells attacking the cells that make insulin. So here we thought, let's see if we can find CD8 cytotoxic T cells that are attacking the cells that make SNX8. And those cells happen to be part of your immune system and part of a part called the myeloid compartment that's all over the body. And so you think of macrophages that are all over in your heart, in your lungs, in different places that are involved. And those are the cells that make SNX8. And through very detailed work that we did along with collaborators at St. Jude's and Paul Thomas and Robert Medelman, we were able to take blood from these patients, isolate individually the T cells that were responding to SNX8, and sequence their receptors so that we could show with absolute certainty that these cells are indeed autoreactive. And we further showed that the T cells were also cross-reactive. So we identified these cytotoxic T cells that are expanded in patients with MIC that target both the part of the nucleoprotein and the part of SNX8 that look the same. And then finally, I would just say that these findings do show some similarity to other autoimmune diseases that are thought to be related to a previous viral infection. And I think a really important finding of ours is that we were able to show on a mechanistic level exactly how to connect a specific infection to an autoimmune process. And so I think that's something that can be built upon going forward.
Well, Aaron, I've got to say that was just the most clear explanation of really what were the specific aims of your study, the approach, and the findings that, when I read the paper, candidly, I had to slow down and read it a couple of times. And you just described it so beautifully. The logical next question I think that comes to mind for me and probably many of those listening is, what are the next research priorities given what your study showed? But before I ask you that, could I ask you both, where has MIS-C gone? As we all remember, in the early months of 2020, while fortunately there weren't a lot of children, the ones that we saw got quite ill rapidly, many of them needing ECMO in a matter of hours. And now it's gone. Where has it gone?
I don't have a complete answer, but I can tell you a few things. One is that it now almost exclusively exists in children who have been unvaccinated. So the CDC recently came out with a report of every case of MIS-C that had been reported in 2023. And the vast, vast majority, more than 80% of them, were in children who had not been vaccinated. And so it seems quite clear that pre-existing immunity, either from natural infection passed on from mother to child during birth or from vaccination, is protective. And if you think about what we found in our study, it makes sense with our findings as well, because if you have preformed anti-spike immunity, you're not going to mount and need to mount the same anti-nucleocapsid protein immune response that ultimately we think links this to MIS-C.
And I'll just add that in many of the prior pandemics, especially those influenza pandemics, when the novel virus emerges that people have no immunity to, there often are rare complications that appear to be hyperinflammatory and somewhat autoimmune, whether it's acute disseminated encephalomyelitis, other types of complications that then go away as the pandemic progresses and it becomes a seasonal virus. So it's not surprising that these types of complications emerge. It was surprising the age range that this peaked in school-age children. So it was very surprising that somewhat narrow age range that of the most of the patients affected, and that's still not completely explained.
Thank you both. This is absolutely fascinating to understand this better, this disorder that we had no name for when it first emerged. And now here we are discussing at this level of detail what could be one of the mechanistic drivers. Given your findings here in this study published in Nature in August of 2024, where do we go next? What are the research priorities?
Thank you for pointing out just how mysterious this disease was at the beginning. And I think when I think about our results here, what I think is really important is that even though we certainly haven't figured everything out, there are huge unanswered questions, which I'll talk about in a moment. We have started to make a fair amount of progress and can now sort of point to specific things that are occurring. And I think it's just important to remember in pediatric critical care, we often encounter patients that have these severe inflammatory diseases that are quite enigmatic. And sometimes it can feel like there just is no way to figure out what's really happening. How can we figure it out? And so I think one really important takeaway from this research is that if you do team science and you build a big collaborative cohort and you collaborate with experts in various areas around the country, that you actually can start to chip away at the mystery. And so I think that's a huge point here. And because of that, my next research priority with MISC is taking it one step further and proving that these T cells and these antibodies, either individually or together, are sufficient to cause a disease that looks like MISC. We can't do it in humans, but we can isolate the patient-derived antibodies, we can isolate the T cells, and we can do some experiments, hopefully in mice, to see if it actually recreates the disease. And if it does, that would mean that we have a platform here that will enable us to begin discovering the links between viral infections, any infection, and subsequent autoimmunity.
Well, Aaron, the way you just described, I think you used the term team science and working across disciplines. And I have to salute you both. I've watched Adrienne develop first the PIC-Flu network that was sitting there ready for COVID, then the overcoming COVID, and all the work that she did allowed the control population, which allowed you to make some of these comparative findings, and is really inspiring. And so it opens the door to my last question. We three are pediatric intensivists, and we well know that across critical care, for too long, we've relied on syndromes. They're helpful clinical constructs, but ARDS, sepsis, that phenotypically, let alone mechanistically, below that umbrella term, were many different patients, and we kept doing trials on syndromes and why we were getting negative trials. And your findings, to me, opened the door to really further understanding and helping to understand what is really perhaps phenotypic differences, mechanistic differences across what appear to be the same type of presentation. So my question is, where do you take this next? Does it have implications for long COVID, which is seen in children, fortunately not as much as in adults? Or does it have even implications, as I just said, for possibly sepsis or ARDS, that this is more than just a post-viral mechanistic understanding you've unlocked? Is there more here, or am I stretching it too far, too fast?
Well, that's a really great question, Jeff, and I'm going to also have Aaron comment on it. But I do think that, especially for some of these patients with long COVID, it's worth investigating whether this is playing a role. And I know that the NIH has a large cohort of patients they're following up and phenotyping and trying to figure out exactly the outcomes as well as the triggers of what's going on. But as far as these acute hyperinflammatory syndromes like sepsis and ARDS, where they continue on after the trigger is gone, I do think that it's worthwhile to investigate whether this is playing a role in some of those disorders. And I know that Aaron is working hard to develop his platform and expand it to also hopefully become a bit more efficient on how we can look at some of these disorders. And I'll let him comment on that.
I echo exactly what you were saying, Adrienne. I think the approach we used in this paper, we could really directly apply to look at long COVID, and that's something we plan to do. With regards to the other types of diseases we've been talking about, sepsis, ARDS, and then some of the ones that are a little more similar to MISC and that they seem to be post-infectious inflammatory syndromes, I think there's a couple of reasons where this work's important. The first I kind of noted earlier, which is that I think this shows that if we really focus and work as a team, we can start to solve these enigmatic processes that I think sometimes can feel overwhelming. And so I think, one, it just means we really should keep looking under the hood and really digging into them, because maybe there's something we'll find. But second is more specific, which is the relationship between infections and autoimmunity, or infections and immune dysregulation, has not been mechanistically sorted out all that well by the scientific community yet. And I think we're on a precipice of that changing for a variety of diseases with a lot of great science that's going on right now. And I think there's new technologies, some I've described here, but others that we continue to develop, that allow us to really look in much higher resolution at what the adaptive immune system is doing. So everything a T cell is targeting, everything a B cell is targeting, everything an antibody is targeting in the viruses and in the host. And I think if we do that, I'm hopeful we can start to find a little bit more connecting some of these specific infections to these dysregulated immune states, and begin to take these syndromes that you're talking about and get molecular diagnoses to specifically explain them in more detail, which I think will also ultimately be needed for us to take the next step in treatment.
Well, I have to tell you both that I have found this to be one of the most hopeful podcasts that I've been a part of in a long time. The really elegant work that was done here by you both is so admirable. Again, what we've been discussing today is molecular mimicry in multisystem inflammatory syndrome in children, published on the 7th of August, 2024 in Nature. And first author, Aaron Bodansky. Aaron, it's just been a privilege to have you here today. You're such a clear thinker, and can't help but note that you're early in your career, so there's a lot of hope to advance critical care over the next 30 years under your guidance. And of course, with my colleague, Adrienne Randolph. And again, as I mentioned earlier, Adrienne had the foresight many decades ago to form large teams, as you said, Aaron, first with influenza, and that platform was the natural springboard for overcoming COVID, and that's led us to where we are today. So it's been a privilege having you both here on Open Pediatrics podcast. Thanks so much.
Thank you.
Thank you so much for having us.
This has been a production of Open Pediatrics. You can find the resources and journal articles referenced in this podcast in the description. We have more podcasts like this one available everywhere you get your podcasts. Visit openpediatrics.org for more information.